Good morning, students. In the last class, we have learnt about plasma membrane and its functions, how plasma membrane is flexible because it shows the semi-permeability in a selective manner, and we could understand the reason behind its flexibility. We have concluded that there are many proteins and lipids present in the structure of plasma membrane. The concentration of proteins and lipids is much more greater than the concentration of carbohydrates in the plasma membrane. Since there is high concentration of proteins and lipids and very low concentration of carbohydrates, plasma membrane is acting like a semi-permeable membrane due to its flexibility. That's what we have learnt. Now let us learn about cell wall. Cell wall, it is a special outer structure present in plant cells, bacterial cells and fungal cells. Cell wall is the special outer covering structure in plant cells, fungal cells and bacterial cells. In these three, the cell wall is present, right? Then, don't they contain plasma membrane? It's not like that. In addition to plasma membrane, the outer covering structure is present in these cells. There is a specific reason behind this. That we will explain at the end of this topic. Let us discuss about the cell wall of plants. Okay, cell wall of plants, which we can easily observe. The plant cell wall is an outer rigid covering structure. Rigid outer covering structure. In addition to plasma membrane. So, where is it present? Inside the plasma membrane or outside the plasma membrane? Cell wall it is present outside the plasma membrane. I already told, cell wall is present in plant cells, fungal cells and bacterial cells. In all these different organisms, is the cell wall made up of same material? No. Cell wall is mainly made up of more amount of carbohydrate. As cell wall is present in different organisms, different carbohydrates are going to be part of the cell wall. Okay, in plant cells, plant cell wall is mainly composed of cellulose cellulose is a carbohydrate, that is, it is polysaccharide. Polysaccharide. Okay, what about bacteria? Bacteria, they contain the cell wall which is made up of NAG and NAM. It is a complex polysaccharide. It is neuroaminoglycolic acid. NAM, neuroaminomuramic acid. Right? So simply these are the acronyms. NAM and NAG. NAG and NAM are acronym for this neuroaminoglycolic acid, neuroaminomuramic acid. In fungi, the cell wall is made up of chitin. It is a mucopolysaccharide. Right. This is how in different organisms, different carbohydrates are used in the construction of a cell wall. Right. Let us discuss more about the plant cell wall. So whatever the NAG, this bacterial cell wall composition, fungal cell wall composition, that is little extra for you, extra material only, right? The cellulose, as we already have discussed, it is a complex polysaccharide, complex carbohydrate, okay, because it is a polymer. Cellulose is a polymer, right? The monomer for this cellulose is glucose only, right? It provides structural strength to the plants. Cellulose is a complex substance or complex polysaccharide which provides structural strength to the plants. It provides structural strength to the plants. Provide structural strength to the plants. So, how does it provide the structural strength to the plants? We'll discuss slowly. Here we are going to learn two things. Okay. They are plasmolysis and Turgor pressure. We are going to learn two things. They are plasmolysis and Turgor pressure. What are these? Plasmolysis and Turgor pressure. When do these cells are going to observe this plasmolysis and Turgor pressure? What are they? Plasmolysis and Turgor pressure. Both are opposite phenomena, almost like. Right. Let us discuss them. Take a plant cell and place it. Take a plant cell. You have taken a plant cell and you have placed it in a beaker that is containing concentrated salt solution, highly concentrated salt solution, concentrated salt solution. After some time, see, if we carefully observe, this is the cell wall and this is the plasma membrane. Here also this is cell wall. This is plasma membrane. What is the difference between here to here? Whatever the cellular contents that are near to the cell wall, when the plant cell is placed in a concentrated salt solution, the cellular contents, they were moved away from the cell wall. The cellular contents were moved away from the cell wall. This process is called plasmolysis. When will this happen? Inside this cell, most of the cellular contents, they are filled with water. When this water, the liquid portion of the cell is called plasma. The liquid portion of the cell is called what? Plasma. It is the liquid. When the cell loses the plasma, the cellular contents that are surrounded by the plasma membrane, okay, they will get shrink. When they shrink, cell wall is rigid in structure. Cell wall is rigid in structure. It will not lose its shape. It will remain like the same boundary. Whereas as the plasma is lost, the entire contents, they will move away from the cell wall. They will move away from the cell wall. This process is called plasmolysis. The process of moving away by the inner cellular contents of a plant cell. Let us make it much more simpler. When a plant cell is placed in concentrated solution, the inner cellular contents will move away from the cell wall. This process is known as plasmolysis. This process is known as plasmolysis. What is plasmolysis? When a plant cell is placed in concentrated solution or hypertonic solution, then the inner cellular contents will move away from the cell wall. This phenomena of moving away of inner cellular contents from cell wall when the cell is placed in hypertonic solution is known as plasmolysis. This can be, so here what happened? Endosmosis has taken place. What is endosmosis? When water moves, exosmosis has taken place. What is exosmosis? When water moves away from the cell. When water moves away from the cell, means from inside to outside, and it is called exosmosis. Exo means outside. Water is moving outside. Exoosmosis. If a cell is getting swollen when it is placed in some solution, there what happened? Endosmosis has taken place. If a cell is getting shrink, there what happens? Exosmosis has taken place. During plasmolysis, which osmosis has taken place? Exosmosis has taken place. Here, exosmosis has taken place. Right. This can be proved by one activity. Plasmolysis can be proved by activity. We will take a thick leaf, okay, a Rio leaf. What do we do with this leaf? We will peel out this leaf. Peel out. And we'll keep some water on a glass slide. Just water. And put this peel. Observe it under microscope. Observe under microscope. Right, we can see it's what's a nucleus, some small dots, green dots, all the things. Okay. Now, in this water, you add concentrated salt or sugar. Right? Concentrated salt or sugar solution. And leave it for some minutes, like 1 or 2 minutes. Again, observe under the same microscope. Whatever previously whatever the things are near to the cells, after adding the salt or sugar solution, you can see all the things, they were moved away from the cell wall. So you can observe this very clearly. Now, take the Rio leaf, put it in boiling water for few minutes. Boiling kills the cells. It kills the cells. Boiling will kill the cells. Now take the peel out from the boiled leaf. Do the same activity. Then after adding sugar or salt solution also, you will not find any exosmosis, because the cells are already dead. Here exosmosis means osmosis. It is not exhibited by the dead cells. That shows that dead cells do not exhibit osmosis. Dead cells do not exhibit osmosis. This is what is happening with the next. Let us discuss about Turgor pressure. What is this Turgor pressure? And why plant cells do have cell wall, fungal cells do have cell wall, bacterial cells do have cell wall. What is its importance? Cell wall, it makes the cells of plants, fungi and bacteria to withstand with the dilute or very dilute or hypotonic external media. Cell wall will make the plant, fungal and bacterial cells to withstand with very dilute or hypotonic external media. When any plant cell is placed in hypotonic media, what happens? Cell will gain water. Endosmosis takes place. So here, these plant, fungal and bacterial cells are more frequently going to have the exposure of this hypotonic media. Then these cells are more prone to swell and then burst. If you keep adding water in a balloon, what will happen? It will burst after some time. After getting swollen, it will burst. The same way, when these cells are in hypotonic media, the cells will keep gaining water. That makes them, continuously expanded. After that, after some time, bursting takes place. Bursting will happen. So, from this bursting problem, the cell wall will protect them. How? When the cell is fully swollen, it is called turgid cell. What it is called? Turgid cell. Fully swollen cell. When the plant cell or bacterial cell or fungal cell is placed in hypotonic media, it starts swelling. Until which point? Until the plasma membrane reaches near or completely attached to the cell wall. Afterwards, this fully turgid cell will start applying pressure on the cell wall so that I can expand more, I can swell more, like that. The cell membrane or plasma membrane of fully turgid cell, it applies some pressure on cell wall. If cell wall, it is very sensitive and says, okay, you can swell, then what it will happen? It will cause bursting of that cell. But what cell wall is doing? Cell wall exerts an equivalent opposite pressure on a fully turgid cell. Since cell wall is exerting the pressure on turgid cell, it is called Turgor pressure. As cell wall is able to exert Turgor pressure, the cells are not going to swell beyond some limit so that they do not get burst, such that these plant, fungal and bacterial cells are prevented from getting burst even when they are exposed to hypotonic media. Right? Which is that. So, the cell walls make these cells to withstand much greater changes in their surrounding medium. If you just keep some animal cell in the same type of medium, what it will happen? Is animal cell is going to not get burst? Definitely not. It is going to get burst. Why? Because there is no cell wall surrounding the plasma membrane of animal cell. So, automatically the animal cell will get over swollen and then it will get burst. That problem is not there with plant, fungal and bacterial cells. So, we have learnt the major structural composition of cell wall is carbohydrate. There are different carbohydrates that are different polymeric carbohydrates are present in plants, fungi and bacteria. Due to the presence of a cell wall, which two phenomena are observed, plasmolysis and Turgor pressure. In plasmolysis, exosmosis has taken place when the plant cell is placed in hypertonic media, so that the cellular contents were moved away from the cell wall. This process is known as plasmolysis, which happens in hypertonic media when the plant cell is placed. And Turgor pressure, when plant cells or bacterial cells or fungal cells are placed in hypotonic media, the cells will get fully swollen or fully turgid, and the cell wall will exert an equivalent opposite pressure on fully turgid cell, keeping the cell or preventing the cell from not getting burst. This is called Turgor pressure. Because of the ability of the cell wall, the plants, fungal and bacterial cells are prevented from getting burst, are able to withstand much greater changes from their surrounding media. Regarding nucleus, cytoplasm, we will discuss in the coming class. Thank you.